Hello everyone, thank you very much for visiting my poster. My name is Maryam Nazarieh and I will talk about identification of key elements maintaining network equilibrium. A gene is expressed if a transcription factor binds to its promoter or enhancer. Methylation of the transcription factor binding site can suppress the transcription by preventing binding of the transcription factor. In addition to DNA methylation, histone modifications also regulate the gene expression by controlling the chromatin accessibility, which cause no transcription if regulatory elements are not accessible. Therefore, cellular processes such as cellular identity is directed by epigenetic marks of a network of epigenetic and transcriptional cooperation. In cancer, alterations in epigenetic modifications affect several processes such as cell proliferation, apoptosis, and development. One of the challenges of improving cancer therapies is to identify key tumor elements that grow uncontrollably and escape immune control and maintain the equilibrium state. For this purpose, it is suggested to consider the set of influencers as the candidates in the transcriptional network of blood cells, where the influencers are the target genes that are regulated by multiple transcription factors. The assumption comes from the fact that the top genes and microRNAs related to a cancer play a role in a network either as half-degree nodes, dominators, or connectors in the dominating set or minimum connected dominating set. Therefore, the target of these genes, which are regulated by multiple transcription factors, can be considered as the candidates for this problem. Here I have displayed the set of influencers and connectors by red and blue colors as self-weight biomarkers and as the nodes which maintain the network equilibrium. One of the challenges of improving cancer therapies is to identify key tumor elements that grow uncontrollably and escape immune control and maintain the equilibrium state. The target of these genes, which are regulated by multiple transcription factors, can be considered as candidates for this problem. Here I have shown the target gene, which is regulated by 6 transcription factors as influencers in this small network. More than 15% of cancers are caused by viruses which target mainly liver, cervix, stomach, head, and neck, and the problem becomes severe in children whose organs are developing. And calcium plays an important role in growing the bones, especially in children. An infection which incurs immune system deficiency is one of the factors which is caused by little calcium in the blood cells. One of the effects of virus infection is brain disorder, which can be checked at different levels, starting with likelihood when the distance between illusion and reality becomes highlighted, and it can be checked also at a sequence level, as I have mentioned here. The viral infection can affect brain, incurring memory loss, disability to do personal activities, and so on. It indicates that high distance between reality and illusion can be a sign of brain disorder. Since memory CD8 T cells control systematic secondary infections and can protect from chronic infection and cancer as a result of their stem cell-like capacity to expand, it is important to reinforce the immune system cells. Here it is suggested to use the wearable devices which help to check the person's activity in order to compare with the number that the brain has counted. This comparison helps to check the brain health and correct it over time in case of inequality by empowering the brain. One of the immune cells is CD8 T cells, which eliminate infected cells or transform tumor cells. And it is very important to know about the molecular machinery that regulates the efficiency of calcium-dependent target cell killing. Here in this paper, we have studied the role of connector nodes as candidates which regulate calcium-dependent CD8 T cell cytotoxicity. To reduce the distance between reality and illusion, which are caused by the set of influencer nodes, it is suggested to rely on wearable devices through measuring the difference between digital and manual counting. For example, between the number of words that we hear of the song and the actual number of words that are pronounced. Thank you very much for your attention.